Good evening. Welcome back. Tonight, we're staying close to the ground. No ocean, no deep time pressure zones, just a damp log on a forest floor in Japan and a shallow pond at the edge of a wetland. Both are unremarkable at a glance. Both are, on closer inspection, running computations that rival the engineering of human civilization. Take a slow breath. Let your body settle. We're going somewhere very quiet. Picture a fallen oak log in an autumn forest, somewhere in the mountains west of Tokyo. The air is cool, heavy with the smell of wet bark and leaf litter. Mist is settling over the wood in the dark. If you shine a soft light onto the log's surface, you find something that doesn't immediately make sense. Stretched across the bark is a bright yellow branching network, intricate, geometric, unmistakably deliberate. It looks like a diagram of something, like veins or a map of roads. This is Physarum polycephalum, the many-headed slime mould. It is not a plant, not a fungus, not an animal. It is a protist, a single continuous cell that can grow to the size of a dinner plate, containing millions of nuclei swimming in shared cytoplasm. No internal walls, no brain, no nervous system, no eyes. And yet it navigates the complex, irregular topography of the forest floor with what can only be described as mathematical precision. Physarum expands through continuous spatial evaluation. As the membrane creeps across the bark, it encounters dry patches, moisture gradients, pockets of fungal decay, channels carved by wood-boring insects. Every square micrometer of the cell simultaneously acts as a sensor and as a mechanical engine. There is no central processor receiving data and issuing commands. The computation is the movement itself, distributed across the entire body at once. When the leading edge finds a dead end, a dry patch, a toxic residue, the local region simply deflates, withdrawing its mass toward areas of higher humidity and available nutrients. The organism shifts its shape continuously to match the microscopic contours of its environment, probing the terrain without wasting energy on retreat. As it moves, it leaves something behind, a thin trail of dried slime, composed of high molecular weight polysaccharides, glycoproteins, and calcium ions. This trail is not just a lubricant, it functions as an externalized spatial memory. When Physarum encounters its own dried trail, it registers a reduction in movement velocity, a physical signal to avoid exhausted territory. The organism has no internal storage for navigational history. Instead, it writes that history directly onto the surface of the world and reads it back through the mechanics of movement, memory without neurons. The outer layer of the plasmodium gels into a semi-rigid structure, a protective casing around the fluid interior. This dual-phase architecture, part liquid and part solid simultaneously, allows the organism to remain anchored while keeping its interior in constant motion. It is structurally stable and dynamically alive at the same time. The veins of the yellow network are hollow, flexible tubes. Inside them, the cytoplasm pumps back and forth in a continuous rhythmic cycle, driven by the same proteins, actin and myosin, that power human muscle. The fluid rushes forward for roughly 60 seconds, pauses, then reverses direction and flows back. This oscillating stream is how the organism thinks. When a region of the network contacts a food source, a particle of organic matter on the bark, the local rhythm shifts. The tubes in that area contract more rapidly, pulling a higher volume of cytoplasm toward the resource. When a region encounters something hostile, dry air, a toxic compound, the pumping slows, the fluid retreats, the tube narrows. Every reversal of the stream carries chemical markers synthesized in one part of the cell across the entire body. If the northern edge of the network finds nutrients, the altered hydrostatic pressure signal ripples through every vein, reaching the southern edge and triggering a reallocation of mass. The whole organism updates simultaneously, using nothing but the physics of fluid pressure. The geometry of the tubes governs this system with precision. According to Poiseuille's law, the hydraulic resistance of a fluid-carrying cylinder is inversely proportional to the fourth power of its radius. A small increase in tube diameter produces an exponential drop in resistance, allowing vastly more cytoplasm, nutrients, and signaling molecules to flow through. Physarum exploits this relationship constantly. Tubes experiencing high fluid traffic expand and reinforce their walls with denser cytoskeletal filaments. Tubes sitting idle gradually constrict, their materials reabsorbed and redirected elsewhere. The structural thickness of the veins is a physical record of past success. The thin, collapsed pathways record failure. The organism remembers where it has been, not in a brain, but in the diameter of its own body. The driving force behind this peristaltic pump is an automated chemical oscillator powered by ATP hydrolysis. Motor proteins pull against actin filaments, creating localized contractions that are not perfectly synchronized across the network. This slight phase difference establishes a continuous pressure gradient. Cytoplasm, behaving as a non-Newtonian viscoelastic fluid, flows down the gradient from areas of high contraction toward areas of relative relaxation. When the local chemistry resets, the gradient flips. The fluid reverses. The cycle repeats continuously, preventing stagnation and ensuring that every nucleus in the shared cytoplasm receives the same metabolic updates as every other. In 2010, a team of Japanese and British researchers ran an experiment that became one of the most widely cited demonstrations of biological computation. They placed a culture of Physarum polycephalum on a flat dish at the position corresponding to Tokyo on a map of the Kanto region. Around it, at positions matching the geographical locations of the major surrounding cities, Yokohama, Chiba, Kawasaki, Saitama, they placed small oat flakes. Light was applied to open areas of the dish, since Physarum avoids UV exposure, approximating the natural obstacles of mountains and bays. Then they waited. The slime mold expanded outward in all directions, initially forming a dense, uniform web covering every part of the available space. When its edges reached the oat flakes, the architecture began to change. Redundant paths were selectively dismantled. Traffic concentrated along efficient corridors. Within 26 hours, the network had self-organized into a configuration of thick primary tubes connecting the food sources, supported by thin secondary loops for redundancy. The researchers compared this biological network to the actual Tokyo rail system, designed over decades by teams of civil engineers using supercomputers and economic modeling. The match was close to identical in efficiency, structural resilience, and cost-effectiveness. The slime mold had solved, in under a day, what took human planners generations. The mathematical problem Physarum was solving is known as the Steiner tree problem, finding the shortest possible network that connects a given set of points. It is computationally expensive to solve algorithmically. The slime mold sidesteps the computation entirely by making its physical structure the calculation. The network configuration is not a solution arrived at by reasoning. It is the emergent result of local competition between adjacent fluid tubes, each responding to the volume of cytoplasm passing through it. The governing rule is simple. A tube's conductivity increases proportionally to the flow it carries, minus a decay factor representing the baseline metabolic cost of keeping it open. If flow through a vein drops below a threshold, the decay term dominates and the channel undergoes autophagic digestion. If flow increases, the tube expands. The global optimum emerges from millions of these local decisions running in parallel at the speed of fluid diffusion. The network also builds in resilience. The Tokyo rail system, designed for efficiency, is vulnerable to single-point failures. Block one major line and the grid stalls. Physarum maintains small secondary bypass channels that remain dormant under normal conditions. When a primary vein is severed, the sudden pressure change diverts cytoplasmic flow into the dormant loops within minutes. The system self-repairs without any instruction because the repair is a physical inevitability of the pressure dynamics. When resources on the log are exhausted, Physarum doesn't accelerate its search in panic. It slows down. It consolidates its remaining cytoplasm into dense, hardened nodes called sclerotia. The cell walls thicken. Heat shock proteins and hydrophobins coat the fragile intracellular structures, preventing denaturation of metabolic enzymes. The vast continuous cytoplasm divides into distinct macroscopic compartments, each shielded by a tough outer wall impregnated with melanin-like pigments. The internal contents vitrify, transitioning into an amorphous, non-crystalline solid state. Metabolic expenditure drops to less than 1% of baseline. In this form, Physarum is practically indistinguishable from the debris of the log. It can survive months of desiccation, freezing, and nutrient deprivation. When moisture returns, the sclerotia rehydrate, the gel transitions back to sol, and the network resumes, picking up its computation exactly where it left off. This is not passive dormancy. It is a calculated metabolic decision, survival through structural surrender. The organism does not attempt to maintain homeostasis against impossible conditions. It allows its internal state to match the thermodynamic profile of its environment, spending nothing until conditions justify expenditure. Stillness, treated as a navigational strategy. Physarum has been doing this for at least 500 million years. It has outlasted the Cambrian explosion, multiple mass extinctions, the rise and fall of every organism with a spine. Not by competing, but by persisting, running the same efficient calculation on the same damp wood, decade after decade, epoch after epoch. Follow the slope of the land downward from the forest, where mountain runoff trickles through moss and collects into a slow stream. Follow the stream to where it widens into a sheltered freshwater wetland, dense with cattails and water lilies, the surface perfectly still. Submerge your perspective below the waterline. The aquatic world operates on different physical terms. The air-water interface above filters out thermal fluctuation, wind shear, and UV. Below the surface, the water column acts as a natural prism, scattering faint starlight into a diffuse velvet dark glow. Suspended particles of organic matter, fragments of fallen leaves, broken diatom shells, ancient silt, hang motionless in the water column, buoyed by the same density that holds them. At the benthic boundary layer, just above the sediment, gravity is nearly neutralized by buoyancy. For a large single-celled organism, this is a liberation. The cell can expand its membrane outward in 3 dimensions without the danger of collapsing under its own weight. Internal form is determined entirely by cytoplasmic force and local water chemistry. The environment imposes almost no structural constraint. This is where chaos Carolinense lives. The giant amoeba, named by 18th century naturalists who, looking at it through early microscopes, saw its form shifting so continuously and so improbably that they could only describe it as primordial disorder, chaos, after the formless void before creation. It is up to 5 millimeters long, visible to the naked eye as a pale translucent speck, and it operates on precisely the same computational principles as Physarum: no brain, no central nervous system, no fixed form, only the physics of cytoplasm, membrane, and fluid. Chaos. Carolinense carries up to 1,000 nuclei in a single continuous membrane. No internal walls, no compartments, no borders between regions. The entire organism is a unified fluid, one cytoplasm, shared by every nucleus simultaneously. Its movement is a continuous physical transformation between 2 states. The outer layer, the ectoplasm, is a firm, structured gel. It forms the walls of a tube. The inner core, the endoplasm, is a flowing, pressurized stream. To move forward, the amoeba initiates a localized relaxation at its leading edge. The internal hydrostatic pressure instantly pushes the liquid endoplasm into the zone of lowered resistance, extending a soft forward projection, a pseudopod, a false foot. As the cytoplasm reaches the tip of the extending pseudopod, it contacts the outer boundary and transforms, fountaining outward, cooling, solidifying into gel to extend the tube wall further. At the rear of the cell, the reverse process occurs. Rigid gel liquefies back into fluid stream, feeding the forward momentum. The organism is continuously recycling its own physical state, flowing through its environment by converting its body from solid to liquid and back again, advancing through pure viscoelastic mechanics. This process is driven by actin and myosin, the same contractile proteins in Physarum, the same proteins in human muscle. In the ectoplasmic gel, actin filaments are densely cross-linked into a rigid meshwork. When the amoeba initiates movement, a wave of biochemical signals, driven by shifts in intracellular pH and free magnesium levels, causes specialized capping proteins to sever the actin meshwork at the rear of the cell. The gel instantly dissolves into low viscosity sol. Myosin motors simultaneously contract the sides of the ectoplasmic tube, squeezing the liquefied endoplasm forward like a syringe. The cycle repeats continuously, fluid surging toward the tip, gelating at the front, liquefying at the rear, in an infinite loop of self-propelled relocation. Without a nervous system, Carolinense performs environmental calculations through direct physical contact. When a pseudopod encounters something favorable, trace sugars from a bacterial colony, the warmth of a sunlit patch, surface receptors trigger a localized influx of calcium ions. This shifts the cross-linking of the structural proteins in the gel layer, immediately lowering mechanical resistance in that direction. The cytoplasm surges toward the stimulus, widening the path and drawing the rest of the cell forward. When a pseudopod encounters something hostile, low oxygen, high acidity, the cytoskeletal matrix locks into a dense, unyielding gel, a physical wall. The internal fluid stream diverts into alternative branches. The organism maps its world not by reasoning about it, but by flowing into the spaces that support its life and hardening against those that do not. Hunting illustrates this most clearly. When Carolinense detects a paramecium, a smaller, fast-swimming protozoan, it doesn't chase it. Instead, it sends out multiple thin pseudopods in a wide arc, moving around the prey without making contact. When the circle is complete, the tips fuse, enclosing the paramecium in a fluid-filled pocket, a food vacuole. This coordinated circumvallation emerges from a straightforward physical mechanism. The swimming paramecium generates tiny acoustic vibrations and microcurrents in the water. These mechanical disturbances strike the amoeba's membrane, deflecting pressure-sensitive ion channels in the lipid bilayer. The opening of these channels alters cytoskeletal density locally. Because the membrane is continuous and the internal fluid is incompressible, a pressure change on one side of the organism propagates instantaneously across the entire 5 mm span to the other side. The cytoplasm functions as a hydraulic transmission line, conveying mechanical information at the speed of a pressure wave. The encircling pseudopods don't plan around the paramecium. They respond simultaneously and independently to the same mechanical disturbance, and the encirclement is the inevitable physical result. Once enclosed, the food vacuole is drawn inward along internal microtubule tracts. Lysosomes fuse with the vacuole wall and release digestive enzymes. The broken-down nutrients absorbed directly across the vacuole membrane, carried immediately into the circulating cytoplasmic stream, distributed equally to all 1,000 nuclei. Hunting, digestion, and nutrient transport performed by the same undifferentiated fluid structure. Late at night, When the active hunting subsides, Chaos Carolinense draws in its pseudopods and rounds into a resting form along a decaying leaf. The cytoplasmic stream slows to a barely perceptible pulse. The organism settles as a translucent pearl of living water, perfectly matched to the temperature and chemistry of the pond. The 1,000 nuclei sharing a single membrane confer a specific evolutionary advantage, genetic resilience. In a multicellular organism, a damaged cell must repair itself or die, protecting the surrounding body through isolation. In Chaos Carolinense, damaged nuclei are surrounded by healthy ones sharing the same cytoplasm. Repair proteins synthesized by undamaged nuclei distribute through the open fluid, reaching and restoring localized damage without quarantine or sacrifice. The organism is a living democratic pool of genetic material, insulated from localized failure by the sheer number and proximity of healthy copies. When pond conditions deteriorate severely, high summer heat, dropping water levels, oxygen depletion, the amoeba retracts all pseudopods, drawing its body into a low surface area sphere. It secretes a multi-layered cyst wall of dense cellulose fibers. Inside, the cytoplasm condenses, metabolic rate drops to near zero, and all 1,000 nuclei enter suspension. In this encysted state, the organism can survive complete desiccation, freezing temperatures and mechanical stress for months or years. When the autumn rains return, the cyst wall absorbs moisture, softens and splits. The organism pours back into the water completely unchanged. The same nuclei, the same cytoplasm, the same fluid architecture, as if no time had passed. Physarum polycephalum on its oak log and Chaos Carolinense in its sheltered pond share the same fundamental design philosophy. Both are single cells operating without any centralized command. Both navigate through their environments by converting the physics of their own bodies, fluid pressure, gel-sol transitions, chemical gradients, directly into movement and decision. Neither stores information in a brain. Both write their navigational history into the physical world and read it back through the mechanics of how they move. Both can suspend themselves almost indefinitely when conditions demand it, then resume exactly where they left off when the world becomes hospitable again. They are not primitive organisms waiting to evolve into something more complex. They have had 500 million years to develop neurons if neurons were advantageous. They didn't, because they didn't need to. The fluid mechanics of a single continuous cell, operating with direct physical responsiveness to its environment, turned out to be sufficient for navigating the forest floor, solving network optimization problems, hunting moving prey, and surviving geological epochs. Intelligence, it turns out, is not a property of brains specifically. It is a property of any physical system that processes information about its environment and responds to it adaptively. The slime mold and the giant amoeba do this at the scale of fluid dynamics. The result is a form of embodied computation, calculation without abstraction, navigation without representation, memory without neurons. The forest log is quiet now. The yellow network rests, its primary veins still carrying a slow, rhythmic pulse. The peripheral branches withdrawn into sclerotia, waiting for rain. The pond is still. The giant amoeba has rounded into its resting sphere, pseudopods drawn in, cytoplasm moving in a faint, patient loop, keeping everything equally distributed, equally nourished, equally at rest. Both organisms are doing exactly what they need to do. Not more, not less. Your own biology has been running its own automated calculations since before you were aware of them tonight. The systems that regulate your breathing, your temperature, your slow descent towards sleep, none of them require anything from you right now. They know what to do. The log is still. The pond is dark. The networks are holding. Good night.